Hello, this is Donnie Rooney, and this is my photosynthesis lab. So let's get to it. So first, this is your light reaction cycle. You've got sunlight and water as the input, and this right here is going to be your chloroplast, and what's highlighted is going to be the thylakoid and stroma. Of course, there's a lot more in there, but it's a lot larger just to highlight what's going on here. So in order for light reactions to take place, you're going to need NADP plus and ADP plus phosphate along with the water. And then you're going to have basically three different steps in this part. You got photosystem 2, electron transport chain, and photosystem 1. So, and like I said, this takes place in the thylakoids or the granum. So in photosystem 2, it's a protein complex that uses the oxygen evolving complex to break apart water to get oxygen to be used in electron transport chain as the final electron acceptor. It reduces plastoquinone to plastoquinol, which is then this photosystem 2 also allows plastoquinol to be catalyzed into usable plastocyanin in the electron transport chain. And some interesting things to note is the ferredoxins transfer electrons made in photosystem 1 to ferredoxin NADP reductase to make NADPH. And ferredoxin NADP reductase is responsible for converting the electron accepting NADP plus and H plus to usable NADPH in the Calvin cycle. So just some stuff that's going on here. Of course, you got your thylakoid. The stroma is going to be this empty space, and the thylakoid space is going to be inside of here. Your chlorophyll is really going to be on the membrane of this thylakoid, and specifically chlorophyll A, which is the chlorophyll form that is involved in oxygenic photosynthesis. So the reaction shown here, and like I said, this is the whole chloroplast. And now, quickly, we'll move on to the Calvin cycle. So in the Calvin cycle, your CO2 is what's brought in along with the NADPH and ATP. ATP, of course, made in the electron transport chain, and NADPH made with that NADP reductase. So in the Calvin cycle, it's just going to be a cycle with basically these three main parts that were mentioned in the lab. You've got the ribulose biphosphate that is going to be converted into ribulose biphosphate carboxylase and oxygenase to help catalyze the reactions to make glucose, and the other two products are going to be 3-phosphoglycerate and glyceraldehyde 3-phosphate. Oh, just like in last chapter. So again, this is going to be a chloroplast, and you've got your stroma, which is where all this takes place. And whenever the Calvin cycle goes through, the products that you're going to get are the used up NADP plus and ADP plus phosphate. So it's just and glucose, of course, your energy. So just looking at a wide picture of this, basically you've got the light reactions that's going to create all of your energy, especially with the electron transport chain. Photosystem 2 will create the stuff needed for electron transport chain, which will then make all the ATP necessary for energy creation or glucose creation. And then, or not energy creation, but usable food source, basically. And then the stuff from the electron transport chain of photosynthesis will be sent to photosystem 1 to be recycled to be reused in photosystem 2. And when you've got your energy and NADPH, you send it over into the Calvin cycle and use the carbon dioxide plus all your different minerals, vitamins, etc. in these plants or cyanobacteria or whatever is photosynthesizing. And you're going to get glucose. It's going to use up the ATP for ADP plus phosphate, and it's going to use up the NADPH and leave you with NADP plus. And that is the Calvin cycle and light reactions. Thank you. Oh, and also, before I continue, so in the electron transport chain of the light reactions, so a couple things happen. The important ones to this lab are the cytochrome B6F complex transfers the electrons, the E-, to create the protein gradient, and the H+, which is now on the outside of, then this is going on the thylakoid membrane. The H+ is going to get used in the ATP synthase to make ATP, which is basically the same thing as cellular respiration. Of course, it's a lot different, a lot of different stuff used, but in the end, you're doing the same thing. You're using the electron gradient ATP synthase to make your ATP and get your energy. And that's your cycles. Thank you.